Hi, everybody. This is the second lecture for the first week. Again, I will start with my learning objectives. In this particular part of the first week, I would like you by the end of it to be able to describe at least 3 ways that viruses are used to treat health problems. So although we think of viruses as pathogens and causing disease, we have actually been able to utilize viruses in a positive way to help treat health problems. I also would like you to be able to identify some important historical and current viral epidemics and pandemics. This will help kind of set us up for the class of how we went about learning about viruses. Vocabulary for this particular mini lecture includes bacteriophage, gene therapy, vaccines, and emerging viral diseases. So some of the way that we can utilize viruses in health or medicine, there's a number of different ways, and many of these I will get into in much more detail in later chapters. But I want to introduce them as kind of keeping in mind that viruses, although they're pathogens, they can be very useful to scientists. So one of the first ways we can think about utilizing viruses is a particular type of virus called a bacteriophage. And I'm going to jump over to my whiteboard to write out that what a bacteriophage is. So you have that vocabulary word. So a bacteriophage, and I want to break that down. Bacterio refers to bacteria, and phage means to eat. So that might sound a little bit weird, but bacteriophages are actually viruses that infect and destroy bacterial cells. And they're specific for bacterial cells, so they don't do damage to human cells. So keeping that in mind, that bacteriophages are viruses that can infect and destroy bacteria, you can imagine that we might have a use for that. And so I'm going to go back and to my slides. So with bacteriophage therapy, we can use bacteriophage, or viruses that kill bacteria, to actually treat bacterial infections by superbugs, bacteria that are resistant to antibiotics. We can use viruses that will kill those bacteria instead of drugs. And so you can kind of think about it as the enemy of our enemy could become our friend. They can also be used to treat something called bacterial biofilms. Bacteria have an ability to form kind of communities on structures within humans, and they have a protective kind of coating that protects those innermost bacteria. We can actually use bacteriophage to go in and kill the bacteria. Because they're specific for bacteria, they should not cause any damage to our cells or host cells. Another way that we can use viruses is to do gene therapy. So we can often use viruses to actually deliver functional copies of genes that are not working in a host cell. And these viruses can go and basically replace the dysfunctional gene in the host cell with a functioning copy, and therefore cure whatever it is that we're looking at curing. And we'll get into that in more detail as well later in the semester. Another way that we use viruses, and this is probably the way that most of you are familiar with, is that we use them to make vaccines. So Edward Jenner is one of the first to have done something with this. He actually worked with cowpox in order to make a vaccination for smallpox. And we have utilized the research of how vaccines were first developed to now do many other vaccines. And we'll talk about that as we get into the semester. The other thing that we know is that some viruses can actually cause cancer in their hosts. And so we can actually use something called virotherapy that can actually use some of these viruses to kill those cancer cells. And this is probably going to be a little bit just of an overview until we get into that chapter. But we do know that virus infections are linked to up to 12% of all cancers are actually caused by viral infection. And so some that you might have heard of are the idea that HPV can actually cause cervical cancer. With virotherapy, we're actually looking at these what are called oncolytic viruses that basically, again, thinking of these breaking down, lytic means to lyse or to break apart a cancer cell, onco meaning cancer. So we can use these viruses to actually target cells that are cancerous and break them down. The last part of this that I really want to talk about is kind of the history of virology and really talking about how viruses have affected the history of humans through epidemics and pandemics. And so just to kind of give us some background, one of the main pandemics that we think of in the last just over 100 years is the influenza pandemic of 1918. This was termed the Spanish flu, which I'll talk about later as being kind of a misnomer. It was not actually likely started in Spain, but the Spanish flu actually killed between 20 and 50 million people. And it was a very unique viral pneumonia in that it caused a very severe pneumonia that ended up, as you can see, being quite lethal. We have a few others that I'd like you to be familiar with, and we'll get into these again in more detail in the semester. Poliomyelitis, which causes polio. We had an outbreak in 1916, but really where we saw the largest outbreaks were the 1940s and 50s. This one has a mode of transmission that it's something that's been contaminated with human feces. We often saw it in public swimming pools. So it went the route of what is called fecal oral, meaning that there's something that's contaminated that is then ingested. But at the time, we didn't know that's how it was spread. And I'll talk about some of the theories that first came out and how we actually figured out how it was being spread. One that's a little bit more recent is acquired immunodeficiency syndrome, which started in the 1980s that we saw. And this is caused by the retrovirus termed human immunodeficiency virus. At this point, we do not have a vaccine that is available to prevent HIV. And this one is transmitted through bodily fluids. So it actually takes quite close contact. Generally, we think of it as sexually transmitted or through IV drug use. Another interesting one that many of you probably are familiar with was the influenza A. It's termed H1N1 virus of 2009. This was our first influenza pandemic of the 21st century. And in this case, because of the outbreak, what we started to look at was more public health initiatives and education going out to the public of how to do prevention. And we'll talk a lot about that as well in this class, about the idea of public health playing a role with virology. We did have a SARS virus, which was a coronavirus before what we're going through now. So this one is the original SARS. So SARS between 2002 and 2003, that just stands for severe acute respiratory syndrome. And this emerged in China in the early 2000s. And what we saw is that it spread very rapidly in healthcare settings among the workers, patients, and visitors. In this case, it had a slightly different transmission than what we see with SARS-CoV-2 that we're dealing with now. This one was droplet transmission, so it was not airborne, but it did have something that we see with SARS-CoV-2, which is a longer incubation period than many other viral diseases, which meant people were traveling before their symptoms arose, and so they were able to spread it further because they were asymptomatic or did not have symptoms. About 10 years later, we had the Middle East respiratory syndrome. This would be the third of the coronaviruses that the second really, with ours being the third that we saw. This one was named Middle East because it was found in the Middle East. That's where it first originated. We do not know exactly where or what it originated from, but it is suspected that it's actually camels that were the possible animal reservoir, meaning that the virus is typically found in camels, and it made the jump to humans. The last couple are ones that we see as more recent outbreaks and that you're probably familiar with. One of those is measles. Measles, its mode of transmission is airborne, meaning that it can stay in the air, the virus particles, for many hours at a time. It had been eliminated in 2000 in the United States, and now what we're seeing is that it's on the rise. You can probably start to think about reasons why it may have been eliminated and has come back. Primarily what we're seeing is it's a failure of vaccination. So unvaccinated US residents often contracted the disease when they were out of the United States and then came back. Because we've had a decreased vaccination rate kind of from the late 90s, early 2000s, and this was due to a fraudulent report that was linking MMR vaccine to autism. What we've seen is since that paper came out, even though it's been retracted and that physician has lost his medical license, that stigma with vaccines has carried on. And we'll talk about that as well as we get into vaccines, because we see an impact of that on SARS-CoV-2. What I would say is that these recent outbreaks, especially of measles and SARS-CoV-2, tell a tale of kind of the importance of having viral immunity. And we can do that often controlling it through vaccination. And we'll get into that in the semester as well. Some other interesting outbreaks, the Ebola virus disease that we've seen fairly well contained in West Africa. We've had a number of different outbreaks between 2014 and 2016. It's believed that bats are the animal reservoir, so the virus is naturally found in bats, and that it periodically can make that jump into humans and then spread human to human. It spreads very rapidly through healthcare. So when somebody falls ill with this, we see that that can cause an outbreak within the healthcare facility, but also in travel. That's how we've seen some cases spread. We've had about almost 30,000 cases, and almost half of those who were infected died as of February 3rd, 2016. So Ebola virus is highly lethal. And then the last outbreak that I'm going to talk about, and obviously we'll spend some time on this in this class, but I do want you to think about what we're presently living through, whether or not it feels like we are in a pandemic. We still are, because it is unpredictable when we will see increases of cases. So the SARS-CoV-2 pandemic started in 2019 and it continues into today. It spreads rapidly, and there's a couple of reasons. It's asymptomatic in many people, and many people are asymptomatic early on when they can still spread it. In addition, it has a fairly long incubation period, sometimes up to a week now. Early on, it had a little bit longer incubation. We see with newer strains, they've shortened up somewhat. It is airborne transmission, so very similar to measles, meaning that it's extremely contagious because these virus particles can remain in the air for hours on end. At this point, we've had close to 800 million cases, and this is as of December this last year, and close to 7 million deaths. I think I'll need to go back and check, but I think we actually have hit the 7 million death mark as of the start of this course. Vaccines and treatments are available for it, and so it is one that we had a very quick turnaround on the vaccines and treatment, which we'll cover later in the semester as well. This brings us to our last slide of checking in. Again, you can go ahead and pause the video, and then I'll come back and go through the questions. So in my checking in, the first question I have is to name 2 ways that viruses can be utilized by humans. One is bacteriophage therapy, so using viruses to attack bacteria that cause infections in hosts. We can also do gene therapy with viruses, so we can use viruses to carry genes in and replace dysfunctional genes. We can use viruses to make vaccines. And lastly, we can use viruses to attack cancerous cells. Number two, which historical viral outbreak is caught by the fecal oral transmission? That's going to be the poliomyelitis virus. So polio is caught in that manner or transmitted in that fashion. Which virus has camels as a likely reservoir? That's going to be the Middle Eastern Respiratory Syndrome, or MERS. And then the last question is, name two viruses that are spread through airborne transmission. Those would be measles and SARS-CoV-2. And with that, that's the end of our second mini lecture for week 1. Please contact me if you have any questions.